pleased to be here as a representative of imec, as we work with SilTerra, both for this symposium and in general. I think it's a very strong lineup of speakers, and I hope that for our part, we will successfully convey the synergy that we see between life sciences and semiconductors and the spectrum of opportunities that I certainly believe exist when you put these two elements together. So for some of you, imec may be a new institute, so let me say a few words about imec itself. As you heard, we're an R&D institute based in Belgium, historically focused on semiconductor R&D. So over time, we have built up a strong staff of scientists and engineers and have two R&D fabs, so 200 millimeter and 300 millimeter. Over the years, we've expanded to include also healthcare, IoT, energy, all areas where we see this synergy between nanoelectronics and application space, and where the connection of the two really leads to new technological opportunities. Our focus is in industry partnerships, so we sit a little bit between academia and industry. Today, we work with over 500 companies in various areas of the nanoelectronics field, but we do in some cases also do our own spin-offs or help to incubate startup companies. I think the element that's important in all of this is something that Eng Tong already raised, which is that you need an ecosystem for doing this kind of work. There's no way a single institute on its own can successfully bring all this new technology to market or even to the research community. So imec has historically worked very closely with many partners in the semiconductor space, certainly the big foundries, and of course we're here because we have a strong partnership with SilTerra as well, in the life sciences space, but also in other technology domains. This by itself, however, is not enough to bring new technology to the market. We have to work with the design houses, again, as Eng Tong already raised, with suppliers, companies supplying materials, tools, which in many cases do have to be customized if you want to realize new technology. And of course, the application partners, who finally are the ones that understand really the user requirements, the application needs, and who ultimately will bring the new products to the market. So it's this spectrum that I think has helped to propagate the semiconductor industry. And I think imec has been proud to be part of that and proud to see how fast technology has evolved. Really in one lifetime, we've gone from computers that fill a whole room to computers that are 1 billion times more powerful and that fit in your pocket. There are really not many other examples of industries that have gone through that kind of rapid evolution in such a short period of time. So from our side, from imec as an R&D institute, we asked the very naive question of, can you do the same thing in any other domain? The one, of course, I'm here talking about is healthcare, life sciences. What is the equivalent of making 1 billion times improvement in healthcare? And can something like silicon technology play a role in that? So we looked at it quite naively, as engineers do. We tend to measure things. So we measured all the things that we have in life sciences, the antibodies, as we heard about, the cells, bacteria, DNA, and we realized we can make things that are about that size. This by itself doesn't solve any problem. It's just the realization that we can make things about that size. It must be useful in some way. It's not only the size that's relevant, it's also the scale. Biological systems are very complex. You think about the human brain with hundreds of billions of neurons. If you want to understand the human brain, you would naively suggest, well, why don't we use a technology that can handle that kind of complexity? And there are not too many out there. But on a single wafer, you can think about hundreds of billions of transistors. Again, by itself, it doesn't solve any problem. But it is this element of scale where to understand a complex system like a biological system, we thought, why don't we use a set of tools that actually match that scale? So we have the toolbox, but what's the problem? What exactly is it that we should do? So imec, over the last 10 years and perhaps longer, has really invested in trying to build up life sciences capabilities in-house. So we have our historical expertise in process technology, semiconductor technologies. We brought in cell biologists, molecular biologists, surface chemists, and we took the rather naive approach that if they have lunch together, some kind of idea should come out of it. It's the type of long-term vision that perhaps an R&D institute can take, perhaps a company cannot. But today, we have over 100 people working on joint projects that bridge this chasm between technology and life sciences. And what I want to show you through the rest of the presentation are some of the examples of synergies that we have found and technologies that we have developed, some of them that we also do together with our partners here at SilTerra. So let me start with a few devices for molecular analysis. And you'll see through the talk that I have chosen applications where we go up in terms of biological complexity. So first, let's talk about detecting molecules, and why would we want to? One of the areas where it's very important is actually in infectious disease management. Today, infectious diseases are still one of the primary challenges for the healthcare system. It includes things like malaria, like TB, influenza. In Southeast Asia, you certainly have challenges with dengue fever. Today, trying to manage these kind of infectious diseases, manage the outbreaks, becoming more and more difficult. People travel very easily between places. You have densely populated cities where these sorts of diseases can easily spread. So being able to detect the characteristic markers associated with these kinds of diseases becomes more and more critical. Oops. Go forward. What we see when we look at the healthcare space is really two paradigms. In developed countries, you have advanced equipment in very nice labs. You have all the infrastructure needed to run that equipment. In underdeveloped regions, you have very different spectrum. Right? This is a mobile health clinic in Kenya. So they take everything on a camel, they go out to the village, they deliver health care. What's interesting in looking at this is it's actually the one on the right that is point of care. The one on the left is definitely not. You have to go there. But this is the spectrum that you need to face when thinking about the challenges associated with healthcare delivery. So in looking at those pictures, one of the things that struck us as an R&D institute is you often need miniaturization. Miniaturization gives you two benefits. One is it allows you to bring instrumentation closer to the patient. So instead of the patient going to the hospital, perhaps you can bring that technology to the patient. It also allows that camel that's wandering around the desert in Kenya to bring all of those devices to the places that actually need them. So it is, to some extent, about democratization of healthcare. One of the key technologies that we often see in life sciences instrumentation is optics. Medical practitioners, clinicians, typically they want to see things, right? That's how they make a diagnosis. So it could be with cameras, spectrometers, fluorescent detection systems. In all cases, though, it is about manipulating light. If you do that with glass optics, it tends to be bulky. If you can do it on a chip, you can make things much more compact. So this is really leveraging a lot of the photonics technologies that have been developed for the communications industry, but now repurposing it for applications in life sciences. This is about seeing and detecting what you want to see. There's also the element of dealing with the sample itself. So, as you heard, we are mostly water. If you want to diagnose anything, you typically have to deal with some kind of liquid sample. So for that, if you want to go from big instrumentation to compact devices, you need to deal with very basic things. How do you mix samples on a chip? How do you isolate a sample of interest? How do you drive a biochemical reaction? And these are also key building blocks that we saw as being fundamental to trying to democratize these life sciences tools that exist today. So with these two as example building block technologies, I want to show you a few systems that we actually built. So this was a project we did some years ago with Panasonic R&D, actually. And their interest was in doing a rapid genetic test, so what we call a SNP test, single nucleotide polymorphism. The paradigm as they saw it was that if you want to get a genetic test, you go to your doctor, you go to the clinician, they will take a sample, that's sent to a central lab, processed, you come back a few days later and get the result. So the question is, can you get the result when you're there with the doctor the first time, rather than going back a second or third time, rather than having a central lab somewhere else processing the sample? So this is a chip we developed together with them, which works from whole blood. It performs a whole sequence of biochemical reactions to actually give you the result, not within days or hours, but within 3 minutes. So then you will have the result right there when you're with the doctor. They can immediately prescribe a course of treatment if it's needed. So it's about getting the right information at the right time so the doctor can make the right judgment. From the imec side, we focused, of course, on the silicon, on the design of the fluidics, on the process technology for building these devices. Panasonic had the other elements of the ecosystem, some on-chip pumps, some electrochemical sensors, and it was with the combination of two that we could actually build a solution for this space. So this is something that Panasonic is pursuing from their commercial arm, and we hope technology like this will become standard in the future. Another example of a photonics-based technology is one that we developed with Jena Light. So this is a company based in Southern California, San Diego. And in this case, they're using, again, a silicon photonics device. That's the small chip that you see here. And it's based on the response of silicon waveguides to different molecules that bind to them. And it allows for very rapid detection of particular biomolecules and assessment of the concentration. So again, this is something they are doing for infectious diseases, but also other areas like cancer diagnostics, like immunogenicity. I think what was very interesting in the Jena Light case is a few years ago when we had the Ebola outbreak in Western Africa, they were able to develop a test on this platform that could give you a diagnosis within 15 minutes. So if you remember from the news stories of that outbreak, it was very challenging to try and track that outbreak, track the origins, because people would see their doctor, walk from their village for an hour, go back, very difficult to track them again. But if you can immediately have an answer as to whether they're Ebola positive, you can do a lot more to manage those kind of outbreaks. And then finally, I'll mention that we also had a collaboration with our partner, Pacific Biosciences. This was a partnership that we started in 2012, and we're very happy to work with them on some of the new generations of their product. But as Annette is here to talk about it, I think I will let her share with you all the interesting technologies that they were pursuing for their sequencing applications. So that's from the molecular side. Now let me go one step up in biological complexity and talk about individual cells. So there are a number of technologies out there for cell-based analysis. The one I'm going to talk about is cytometry. So this is essentially a cell counting application. You take a sample, you count or analyze every single cell in that sample. It is difficult to handle high volumes of samples. It is something people use as a research tool. It's typically a benchtop system. It's now getting more and more interesting for applications like prenatal screening, where you're looking for fetal cells in a maternal sample, but also for things like cancer diagnostics. So the imec vision is, can you take that big instrument, can you put it on a chip? And then if you want 10x more throughput, instead of buying 10 instruments, you just put 10 components on a chip. Why we're interested in it in the spectrum of cancer diagnostics is that for cancer, we often see that the cancer spreads through the body, through the blood system. Right? That is our body's interconnect system. All our organs are connected in that way. And it's usually the nascent stages of metastasis where you see cancer spreading to sometimes vital organs. The challenge is twofold here. So if you want to do early diagnosis of cancer, you can sometimes do it by analyzing blood samples. There you are typically looking for very rare cells, so few cells in tens of billions. Also very useful for treatment follow-up. So patient who's already been identified as having cancer is going through a therapy. For you to monitor, is that therapy effective? This is one of the tools that you can use if you can really detect these cancerous cells in their blood samples. So the technology that imec has been working on is a microfluidic-based approach. In this case, there are really two elements that I think are quite interesting. So the microfluidics itself is something that we can put on a chip. It's easy to scale. Here I'm showing you a central channel through which we stream blood cells one at a time. The other interesting technology relates to how you detect and then isolate the cells of interest. So we actually image or fluorescently register every cell that passes through the chip. So having detected a cell of interest, you then need to, in our case, deflect it into another chamber so you can do your downstream analysis. We appropriated the technology from inkjet printers. So they have little bubble generators on them, and that's what you see as this small array. By heating up these resistive heaters, you generate very short-lived steam bubbles that can actually deflect a cell into the channel of interest. What's interesting about this is these steam bubbles you can generate in microsecond timescales. There are actually not many actuators you can integrate on a chip that can operate at that kind of speed. And that's why this is a rather unique technology. Not only can we parallelize it on a chip, but we can actually operate at the type of speeds that are necessary to sort through millions or even billions of cells. So for now, we've been focused on looking for the healthy cells. Those ones you have to detect if you're going to find the abnormal ones. So with a single channel, we process up to 5,000 cells per second with purity in the 98 to 99% range. Already getting comparable to a benchtop system, and this is just with a single channel. Of course, the idea is finally to look for the abnormal cells, and that's what we have started doing. So these are data where we spike in tumor cells into a healthy sample, and we start to see, can we isolate out these rare cells? There is a long way to go before this becomes a commercial technology. So this is certainly closer to research phase than commercialization phase. But we think we're really working on the right building blocks to change the paradigm from a large benchtop tool to an integrated compact device. Now let me go one step further, talk about what I call in vitro systems. So this is now collections of cells, tissues, cell cultures. This is a very typical in vitro system. You have huge vats where you grow cells. These can be grown for things like vaccine production. In Belgium, we use this to grow beer. But you can do any number of things. The challenge with these sorts of systems is the monitoring. So you can typically monitor things like temperature, pH, maybe some of the gases. But if you lose a batch, you really lose the whole batch. And the sensor technology needed to monitor some of these things is actually quite primitive in some sense. This becomes especially important as we move to new application domains in things like regenerative medicine, things like cell therapy for cancer, bone regrowth. This is where you have very high value cell cultures, where any risk of contamination is a direct risk for the patient. So you need to be able to monitor very closely what is happening in those kind of systems. You don't need to make them necessarily this big. They can be much more compact, but you have to be sure about what type of cell you're actually producing. So I want to show you one example of a technology we've been working on for this space. And it's a technology for measuring the electrical response of cells. This is what electrical engineers often do. So what I'm showing you here is a microfluidic well plate. The format is probably something familiar to biologists. We have integrated a CMOS chip with that. So it's what we call a multi-electrode array. And it's a chip with a series of electrodes and electronic readout to be able to look at the electrical response of cells like cardiac cells, neuronal cells, where the specific electrical functionality is really part of the cell behavior. So why do you want to do something like this? I'll show you one example. In this example, we have cultured cardiac cells on our chip in these well plates. And then we first measure the electrical response of these cells just in the medium. So we're not interrogating a single cell, we're just looking in the medium. And in fact, you can see a characteristic response, a beating of the cell. That's what our heart does. We then looked at the response we get from the chip itself. And the chip is able to measure from individual cells in that sample. And first of all, you get a much larger signal, but you also get much more detail in terms of the nature of that signal. So why is it important in the case of cardiac cells? It's important because you can start looking at how drugs affect those cells. And in the pharmaceutical world, when a drug is pulled from the market for safety reasons, it's often pulled because of some kind of adverse cardiac response. So that's already being detected after going through all of the clinical trials, the €1 billion investment to actually put that drug on the market. We have shown with a number of drugs that have been pulled for the market that you can already see at a single cell level that the cardiac response is going to be different with that drug. So it's something that we see as a quite powerful tool in toxicology, toxicity assessment, and actually hopefully for other areas in cell culture analysis. So the last topic I want to discuss is in vivo devices. So now this is the full complexity, dealing with a living system. I'll show you two examples. One is a device we have developed for neuroscience research. You can actually see this in the exhibit hall next door. So it's something we published in Nature last year together with our partners from HHMI, from Wellcome Trust. And this is a CMOS chip for doing neural recording in animal models. So it's for research use only. The idea was, can you take all of the electronics that a neuroscientist typically uses for measuring from a brain, put it onto a single chip? This is actually a little bit misleading because it's not that we took this full rack and put it on the chip. We actually took 16 of these racks and put it on the chip. So this was a comment I was making earlier to one gentleman that you sometimes don't understand from the life sciences perspective how much you can do with a chip. So sometimes we also have to help them say, you know, we can do more than this rack if you want. So for us, this was a really nice partnership with those application institutes, also one where we leveraged SilTerra's CMOS technology to develop the probe, and one where we had to actually deal with a number of challenges. There's obviously the circuit design element, there's the process technology element, there was the full electronic system that we finally had to build around it, and we also had to worry about the biocompatibility of the system. What's interesting is the things that are biocompatible tend not to be the materials you put in a fab. So trying to bridge this gap and find the right spectrum of materials that you can actually use in a living organism was one of the key elements of the project. So this is something that we have released as a beta. It will actually go to market for the entire neuroscience community sometime in Q4 this year. And it is already orders of magnitude more powerful than any device that neuroscientists have today. So for us, it was a really nice connection between technology, application, and system development. So this is for research applications. There are actually extensions of this to therapeutic applications. So this was a project that we had done with DARPA. And DARPA was interested in technologies for artificial limbs. So I'm showing you here Often they have cases where patients, soldiers, they have lost a limb. You have very advanced prosthetics that are now available. But the challenge you often face is you don't have any bidirectional signal going to your brain. So although you have advanced sensors in this prosthetic, your brain is not actually reading that information. So the idea was, could we take something like our neuroprobe technology, which is really about measuring signals from a neuron, and use it to actually help with the peripheral nervous system. So interface directly with nerves in the arm, in the leg, to read signals coming from a prosthetic, but also to transmit signals from the brain. So why is CMOS technology especially interesting for this? It's interesting because these peripheral nerves that run through your limbs, they actually have thousands of little wires, so thousands of little nerves. So if you want to interface that with that, you need a technology with thousands of electrodes, thousands of stimulation sites. You need a technology at the right scale. So that was what we were focused on in this research project. Again, this is one that is certainly in the research phase, not ready to be commercialized yet, but I think it was a nice demonstration of how we could take technology and really address a key need in the area of prosthetics. So those are the examples I wanted to share. Let me come back to this slide that I showed at the beginning and the question of, can technology transform healthcare? I think not by itself, right? Technology is one part of the ecosystem. But I do see that there is something very fundamental from the technology side that we can contribute into the healthcare domain. And I think by working together with all the partners in the ecosystem, I don't know if we'll achieve the billion-X improvement, but I see a lot of opportunity in this space to deliver new solutions for patients ultimately. So with that, thank you very much.